So basically, the two areas of therapeutics is diagnostics. Of nanomedicine, I should say, is diagnostic and therapeutic. And this is really under a realm that people have named nanomedicine, okay? And this is where we have these inorganic magnetic nanoparticles that can do all sorts of wonderful tricks. So under the realm of diagnostics, they can help in enhance imaging. So particularly this is MRI imaging. And under the realm of therapeutics, there's a whole range of ways in which magnetic nanoparticles can be used. I should say I'm looking here predominantly at magnetite. This is where they can be used to steer a sort of a therapy, so a drug may be attached to them, and using a magnetic field, this cartoon here of a mouse is from a Nature review. Using a magnetic field, you can steer the drug in question to a targeted site, say at the site of a tumour. You can also use the magnetic effect of the particle itself, finding something called hyperthermic therapy, or there's now new areas with photothermic therapy, which also work with magnetite. And that's where basically you have an alternating magnetic field, and that will heat up the particle. So you can steer the magnet to the site of the, say, tumour, and then you can apply a heating effect through the magnetic properties they have. So quite powerful stuff if we can use them effectively. For nanotechnology, there's a whole range of different nanotechnological applications for nanoparticles, but the one that will touch on just right at the end as a kind of where we can go next is around data storage. So just as a very light touch general introduction to that, our hard drives, you probably recognise here, are magnetic films, and they store data in the realm in the form of a magnetic dipole, either dipole up or down. There's a bit of information like zeros and ones, so it's sort of that simple. Obviously, it's a bit more complicated about how they're arranged, but basically, as we increase the need for more data, then we want to try and get more information on as small area as possible. So the smaller we can get those particles, and the high definition between those sort of domains of data is really crucial. They seem very different sort of applications, but with both of them, we really need uniformity of size and shape of our particles. That obviously controls crucial for the consistent magnetism, but it's also crucial for the applications. So I've already said about kind of the spacing for the magnetic hard drives, but also size and shape is really important for nanomedicine too. So the body will treat particles differently depending on their size, but also their shape, and actually the effects of the therapy are very much dependent on whether or not a particle has points or is spherical. So those are really critical to be able to control. Also, coating and functionality is important. But in whatever application you want, you really want the ability to be able to tune your particles. Now, going back to the chemistry, now that's why we want magnetic nanoparticles. Let's think about how we might synthesize them. So with the applications, as I said, we need this uniformity, but I've put here next to the uniformity these symbols for basically to remind me to think about the green aspects too, the sustainability. And we really want to be able to produce particles in a way that isn't harmful and isn't toxic to the environment. So the best way to do that is something simple like a room temperature co-precipitation of magnetite. And this is where we get ferrous ions and ferric ions in solution, and we simply make them insoluble. We get them to fall out of solution as the co-precipitate of magnetite by raising the pH. Now that's all very easy, all very sustainable, but what we get is a huge mix of different ion oxides and also different shapes and sizes. So there's not much control over this process. When you start adding control, that's when things like organic solvents and regularly used and different more toxic precursors. So there's a real problem here around being green and having lack of control, and having more control, but having much more environmentally damaging reactions. And this is where basically biomineralization comes in. So this is where we see that nature is able to control the size and shapes of a whole range of minerals, and they can do that under ambient conditions and has done for millennia. In fact, they've been perfecting this over millennia. And this is from the macro scale. We'll see this skeleton here, right down to the nanoscale. You see some nice pictures here of diatoms, which have the most beautiful architectures. But one of my favourites, and my favourite organism I'm working with, of course, apart from humans, of course, and maybe dogs, is this magnetic bacteria. And as you can see here, I haven't actually put a scale bar on, which is poor, but they're about 6 microns long, and they make these magnetite nanoparticles within their cell. And this is where I'm going to show a little video of them, because they are beautiful things. So you can see these are spirula, so they're moving in a corkscrew-like manner, and you can see they're basically pointing all there, and then they've turned now to go down and now across, and that's because they will turn and move with a magnetic field. And as you can imagine, why that is because they take an ion from the environment and they biomineralize it in that row of particles, as you can see here, within their cell. So this is a graphic to try to understand a little bit more about how they do that. So the red dots are basically like ions in solution in the environment, and they take these up, and what they do is they invaginate a membrane from the cytoplasmic membrane, from the inner membrane, and that invagination process recruits lots of biomineralization proteins into the membrane, and these are shown here in green. As you can see here, these are the proteins that will import the ion, and the red protein here that I've shown sort of embedded in the membrane is a classic biomineralization protein, and that might form the site to nucleate the particle forming, or indeed control the shape. There's a whole range of these, a suite, if you like, of biomineralization proteins that exist in this membrane, and that makes a very particular size and shape of particle, as you can see here. So this is really kind of the inspiration for me, because if there's these proteins that can control the formation of exact particles, excuse me, I'll start the video again, then that's basically ideal for our purposes. So what I want to talk about today, I've got basically a whistle-stop tour of 4 areas. I'll be spending the longest on points 1 and 3. So I'm going to look at the magnetosomes themselves, of how magnetic bacteria, we can actually just isolate the magnetosomes and use those for cancer therapies, and how we might enhance them with different forms of either doping the particles themselves or functionalizing them. I'm also going to talk about how we might then mimic those magnetosomes. The third topic is where we consider the fact that those magnetic bacteria have these biomineralization proteins, and how we can use those for a green synthesis, so use the proteins as an additive, but also then learn from those proteins to look at bio-inspired additives from the information we learn from them. And I'm going to finish just on a note about new directions and how we can maybe use some of these methods that I've developed for magnetite for new materials, for new nanomaterials. So hopefully that will fill the time, and we'll see how we go. So let's start with magnetosomes themselves for cancer therapies. So this goes back way back to when I did my master's project, and this actually is what inspired me to stay in this field and work with magnetic bacteria. So I did my master's project with magnetic bacteria, and the plan was to dope them with cobalt. Now I should say it was a very silly plan, and if I'd ever talked to a biologist at the time, they'd have told me it was a silly plan because cobalt's very poisonous, and the magnetic bacteria probably wouldn't want to take it up anyway. And what was I thinking of? However, because I was a chemist, and this is the thing about discipline hopping and being a bit multidisciplinary, I didn't really think of that. So I just thought I'd feed them some iron and some cobalt at various amounts and see what happened. And actually it did work, because there is this, I happened upon, luckily, this window of where you can add enough cobalt not to kill them, but to dope the actual magnetosomes themselves. So they take in the iron and the cobalt, but they incorporate a small amount of cobalt, between 1% and 1% in this paper when I wrote this. But then I've done later publications where we've managed to get up to 3% of cobalt doping. Now that, you may think, what's the point in that? I want to kind of remind you about when I talked about the hypothermic therapy, where you can increase the heat, where you can have heating by putting them in an alternating magnetic field. If we add cobalt, that actually adds a level of coercivity, a level of hardness to the magnet, and that increases the coercivity. So as you can see, hopefully on the right hand side here, the red dotted hysteresis loop is the one with the cobalt doping, and the black in the middle is the control. And that increase in hardness, that fattening of that loop, actually will increase the hypothermic heating. Now this is even more important and interesting because people have proved, not me, but other researchers have shown that magnetosomes actually have a larger hypothermic heating than particles, synthetic particles of a similar size. And hopefully you can see here on the graph on the left that if you just take any magnetic field in these lower regions and go up, the synthetic particles will give this heat loss per cycle, whereas magnetosomes will give basically twice as much. It's not clear why that is, but if we can then dope them with cobalt, then we get an even larger increase in temperature. And indeed, this has been shown by Ala Frendre, who basically did this. They added, they copied the method that I showed with adding the cobalt, and they did see this increase in temperature with adding cobalt. So really, the magnetosomes themselves should be quite powerful nanomedicines. So some of the work that we've done with this is some of the work from Zainab, who was a PhD student in my lab, and she worked heavily with these magnetosomes, doping them with different sorts of metals. She did manganese, cobalt, and copper. And you can see here, this is the control data, and then so this is control, and then basically if we see that they have survived, or there is apoptosis or necrosis. And you can see, hopefully, that when we have magnetosomes and they are subjected to hypothermia, that compared to the control, there is a lot more cell death. And with the cobalt doping, that's the most. And that's obviously because of this anisotropy that increases the heating. She also did a little bit of work in vivo, and you can see actually that the cobalt doped magnetosomes make their way into the tumours. And actually, the tumours don't grow as much when there is the cobalt doped magnetosomes present and there is hypothermic heating. We didn't do enough of this data actually for it to be good enough to publish, but it's really promising starting work. So we've also looked at how we can then enhance these magnetosomes. So we can enhance the core, the magnetic properties of the core, but can we functionalize their surface to do things like I said in that first slide, like attach drugs and the like. So this is a paper that's just recently come out from Zainab and another one of my PhD students who's just submitted their thesis last month, Chris Legg. And basically what you can see here is that he tried to show the different sorts of uptake and toxicity of magnetosomes compared to just bare synthetic magnetite, and then also coated magnetite as well of different sizes. And basically the take-home story is that they're all uptaken, and they're basically generally not toxic unless you're doing something with them, which is all good. But hopefully you can see from this slide that we managed to also decorate those magnetosomes. So here we've got some different fluorescent labels on them. So this is the control at the top where nothing's happening. But here we can see that we've got red fluorescence attached to our magnetosomes and green fluorescence as well. And we've also imaged them in cells. So we've managed to basically enhance the particles by not only changing the core of them by doping them, and we can change the magnetic properties, but we can also attach things. Here we've just shown with different fluorescent molecules, but obviously that could be substituted for a drug or the likes, to show that they also do uptake into cells. More recently, we're doing some work with a colleague of mine, Monita, in oncology, and her postdoc Faith Howard, where the project that we have jointly with them is looking at oncolytic viruses, and these will of course attack cancer, and they are basically an excellent sort of Trojan horse, if you like, to kill cancerous cells. So they will go towards cancerous cells and also destroy them. But we can improve these by decorating them with magnetosomes, so we can actually magnetically target them as well. And some recent work which Faith has just submitted to nanotechnology the other week shows that basically this idea is a success story. So we see that with this cell line, these cancer cells, when we have just the magnetic particles, you know, with magnetic targeting, nothing much happens. You'd expect that because there isn't the virus present. When the virus is present, it doesn't necessarily go to the cancer cells because it has to be pretty near to be able to target to them. When you have both, so you can magnetically target and you have the oncolytic virus, then you get this cell death. The other exciting thing to note is the cancer cell death here is shown after it's been incubated with neutralizing antibodies. And you can see here that the magnetic nanoparticles not only guide the oncolytic virus to the cancer site, but they also seem to protect it from being attacked by antibodies. So this is a really sort of important finding, and we're very excited and hope that it will get published soon, so you can all read about it. Okay, so I'm going to move on now to the second area of magnetosome mimics, and I guess the whole rest of this talk really is based on the idea that although I've just been singing and dancing about how fantastic magnetosomes themselves are as a very important nanomedicine material, they're a bit tricky to kind of work with because the bacteria themselves, they're hard to grow, and they kind of they want to live more than help us out with our medicine. So it's not a high yielding process, I guess is what I'd say. And perhaps with medicine that's kind of worth it, but with other areas of nanotechnology, perhaps we need to be able to make more of these particles at a better rate to be able to manufacture them. So this is now basically, I suppose, looking for solutions where we can learn from nature and take the best bits, but be able to produce them outside of a cell, and to be able to produce them at much higher quantities, but still retaining those advantageous properties. So basically, this is all about if we can mimic them. So sorry, this is a bit of a busy slide. It's kind of looking bigger on my screen than I recall it. But this is where the idea that we could see if we could actually just make a magnetosome. So we can know that the magnetosome is this this lipid vesicle, and then you grow the magnetite within it. And this is not like the first idea, the first time this was proposed. This is actually a part of a paper from Steve Mann and co. from the very early 80s when the magnetic bacteria were first discovered, and he was looking into compartmentalization. This idea that actually we could grow crystals within these liposomes. This is also from that paper, and you can see that there's only a very tiny crystal, and that would be expected because if you imagine you've got your ions in solution, and if you condense those out as a crystal, there's going to be a lot smaller particle because you've only got a finite amount of the metal ions there. So what I've been sort of, I suppose, thinking about is we probably want to mimic nature more so, and think about the fact that that iron, remember the red dots from that cartoon I showed you, the schematic, we want to basically have an infinite influx of iron. So basically we need to reverse this process. We need to have a base interior, and we need to be able to import iron. Okay, so that's kind of my prerequisite. I need to have a lipid or indeed polymer compartment, a vesicle, a basic interior, and find a way to import iron. So the first kind of thing I looked at was the polymerosomes. So I've got a diblock copolymer here to mimic what a membrane looks like. So we've got the hydrophobic interior, the hydrophilic exterior, just like what a lipid would be. We've got a triblock, which now doesn't look like a lipid bilayer. It's got the hydrophilic just on the exterior and one block for the hydrophobic on the middle. And finally, I looked at if we functionalize the diblock, so basically be more akin to how the membrane might look, but with a protein in. Now, how do we transport the iron in? This is where we use the technique of electroporation, which again, any of you cell biologists out there will know for getting DNA into cells. It's a way of opening pores in membranes to let things in. And here we're basically letting in the iron ions. So we tried this first with our simple di-block copolymer, PDB-PEO. And what we formed is not a magnetosome. You'll see here this central image. And we've basically formed almost like a pudding, lots of tiny, tiny particles embedded in the membrane of our vesicles. And if you think about it, that is kind of what you'd expect to get, because if you think about the schematic, the interface where both the iron and the base will meet is in the membrane when the pore is opened up. So that's obviously where the particles would form, and one feels a bit silly for not realising that earlier. Oh, I've lost a slide. Oh well. The slide I've lost basically shows the same story, but with the tri-block, the same sort of like pudding-like tiny little currents in a Christmas pudding type arrangement. So then, I suppose to combat that, but also to think more mimicking wise, we thought we'd try to mimic what the actual membrane looks like with proteins in. So here we basically ensured that we had an interior polymer that had some acidic residues, and that's because a protein, which I'm not going to have time to talk about today, but a nucleating protein called MS6 has lots of acidic residues that are displayed on the interior of the magnetosome membrane. And that's where basically the iron ions can bind and nucleate the formation of magnetite. So we form these polymerosomes, and sure enough, we do get the core magnetite precipitation, as we see more like the magnetosomes themselves. We've also done a lot of work with lipids. This is a collaboration that I had with UCL, a colleague called Mohan Erdesinger and his postdoc, where we basically made a high throughput synthesis, made DSPC lipid vesicles that were filled with sodium hydroxide, and these then dropped into, if you like, a tray of iron solution, and then we could electroporate within the tray. So that was my computer seems to have now broken. Oh, there we are. It's come back again. I must be overheating it. And here we'll see some nice cryo microscopy of the magnetite formed with the membrane around it. So these are really now looking a lot like magnetosomes. So in summary of the kind of the things that we can achieve there, we can see that when we've got liposomes, they're quite similar, I guess, to the magnetosomes, and the polymerosomes actually have very different forms. But if we try to add some more sort of bioinspiration, we get something more similar to a magnetosome. The biological slant on it, the biomedical, I should say, slant on it, is that we have obviously looked at these for both hypothermic and diagnostic MRI applications. And the first materials I showed you, the di-block copolymers, they show very nice and enhanced MRI signals. And they also show very nice hypothermic heating. Similarly, the last ones I showed, the bio-inspired particles, they also show a 5-degree increase in heating over 10 minutes. So really promising particles for therapeutic and diagnostic applications. The third area I want to talk to you about is where we might then look at the proteins that the magnetosomes use to form, so the biomineralization proteins, and see if we can actually gain inspiration to make additives for this process. Okay, so this goes back to some work that we started with way back at the beginning of the fellowship that I began. And this is where we work with this protein MS6, which I said I will not have time to talk about today. I've spent most of my life, it seems, actually working with MS6, but I could do the whole 3 hours on that protein. So I don't want to get distracted with that. I want to talk about more recent things. But what we saw with that is that basically, as I said previously, if you just do a room temperature co-precipitation of magnetite, you get this mess. If you add the MS6 protein, which is the biomimerization protein from the magnetic bacteria, then you get a nice shaped particle, as you can see here. MS6 was found tightly bound to the magnetosome from the membrane, so it was assumed to be important. But since then, there's been a lot of work on the genetics, and it's been shown that it's in a gene cluster next to MSF. And MSF has been shown through, again, more genetic knockout mutagenesis to be really important for controlling size and shape. So we have repeated the work that I did originally with MS6 with MSF. Because the idea, I guess, that you can use a protein as an additive is very attractive, because it's almost getting the best of both worlds. You can have that large scale, very green, sustainable room temperature co-precipitation reaction, which, as I said, is problematic because you have that lack of control. But if you can add protein, then we're now giving control to that synthesis. Okay, so we did, as I said, repeat this with MSF. The findings here, I think, are really interesting. Well, I suppose I would say that. But as you can see, we've got control particles here, and we've got MSF particles here. And from the last slide, it's clear that the MSF has controlled the particles' size and shape. Well, actually, the size is less obvious. Actually, it's the shape, but also the composition. So you can see here the magnetism. This is the MSF, which has the full magnetite pure composition. What I'm showing the comparison, and what's, I suppose, really interesting about this paper is we also looked at two homologs. So MAMF and MMXF are very, very similar proteins to MSF. They just have these very slight differences. So we actually assumed that they'd behave quite similarly. But what we found is that they really, really don't. So hopefully you can see from just the images, the TM images, that the shape and size is very poor. And also, as you can see from the magnetism, the composition is also very poor. And that's fascinating. I hope you can see from the sequences. It's fascinating that so little difference in the sequences actually has that very dramatic effect. And bear with me, because that will come back in a bit. So I should say before I go to the next slide, that one of the problems with these proteins, as I said, it's the best of all worlds, but anyone in the audience here who's worked with membrane proteins will know I'm kind of skirting over some large issues. So both MSF and MS6 that I've worked heavily with are very difficult to work with because they're membrane proteins, particularly MS6 actually. And that's where you've got this amphiphilic protein. It tends to want to fall out of solution. It's not soluble. So actually obtaining it as an additive is not something that's scalable. When I was talking about this sort of dream of making something much greener, much more scalable process, using membrane proteins is not, I guess, the way forward there. So what we can do, I guess, is because we've understood a lot more about how these proteins work, we can take the specific sort of active region that we know more about, which is this top bit, if you like, and the bit that's embedded in the membrane, the stalk, which is the problem. We can kind of maybe just get rid of that and display the active region on some sort of scaffold, and then we can use this newly artificial biomimerization protein, if you like, to add that to a room temperature co-precipitation instead. Okay, so that's basically what we did when we looked at our coiled coil constructs. So this is where we got MMSF and MS13. These are two membrane proteins that display a loop presenting here to nucleate or form or help control the shape of the forming magnetite particle. As I say, these membrane transmembrane regions are the problem. So if we just replace those with a coil, so we're just simply displaying this active loop region, both here and here, onto a coil. We characterized this and saw that indeed it does form the monomer coil rather than a more complex arrangement. And what we found from this is we found that actually the particle shape is very well controlled by the MSF coil. That's compared here to the protein. And what's really interesting is one could say, well, actually, what if just that sequence, that loop region, what if that itself is all that's required? So we did also do the reaction in the room temperature co-precipitation with just the peptide, just the loop region, and that had a diabolical results. So as you can see, the control over particle shape in this graph down the bottom, size is atrocious with the peptide. You get a huge range of sizes and lots of shapes. So it really does need to be constrained in this loop formation, which the transmembrane regions of the protein do in the membrane, and this coil substitutes for very nicely. The MS13 though, actually you'll see we don't get quite as good results for that protein, and we can see here that the magnetite binding is best with the MSF coil, but actually the iron binding is better with the MS13, which leads us to think this is more of a nucleation protein, the MS13. So there's different mechanisms, I guess, in play. Whereas the fact that the MSF coil controls the shape, I suppose means that these monomers are much easier to control within this artificial scaffold. Okay. So when I said before, remember this difference in sequence, what this coil scaffold has allowed us to do, it's allowed us to basically be able to work with MSF and indeed many other membrane proteins, if you like, with much more ease without them being membrane proteins. And this means that we can actually do some very simple and easy mutagenesis on this loop region to try to understand what it is about the MSF coil, which is so much better than the MAMF and the MXF coil. So what we did is we've gradually sort of gone one amino acid at a time from the MSF to the MAMF, and similarly from the MSF to the MXF, one amino acid at a time, changing it and seeing which ones are the crucial amino acids. This isn't published yet, but I'll share the data and show you. It's all gone wonky, which is annoying, but hopefully you'll be able to see what we can see. And hopefully you'll see that the shape control is a really interesting feature, and this is part C here at the bottom of the slide. So the MSF in the middle here has got this fantastic shape control. When we go towards MAMF, so going to the left, you'll see there's this gradual decline. Okay, so it's basically replacing that acidic residue at the beginning that seems to have the most, the acidic residue at the beginning, and both of them together having that effect. Whereas the MXF seems to be all over the place, and that I think is quite fascinating. And this is where we see that basically we have this huge loss of shape control when we replace. I'm going to bring these up to kind of explain my point. When we replace this this aromatic group here, I know that not all of you will be amino acid specialists, so I'm going to try and talk through this more friendly. So we've got basically the amino acid, which is aromatic, and when we replace the aromatic with, I'm getting confused now here, with this non-aromatic, so going from the top here, we've got this aromatic group right at the very top, and we replace that with something that isn't aromatic. That's when we get that big loss of shape control. Interestingly though, it's recovered when we replace the aromatic now at the top left. We've replaced that with one now with an OH on it, so it's a bit more polar. And maybe that polarity can now interact with this other group that isn't aromatic but polar. So, and this is just a suggestion, maybe the first MSF, those two aromatic groups can interact maybe by some pi interaction, some aromatic stacking. Similarly, maybe this last example at the bottom can interact through some perhaps hydrogen bonding, whereas this middle group definitely cannot interact with each other. So to us, that makes us feel that perhaps this yellow region it's really important that it's held structurally in one sort of conformation, and maybe that's important for the first region, which is more acidic, to be able to work its action. So we've got new PhD students starting to work on this, to look at the structure using NMR of how this this peptide region actually changes shape with these mutations. Okay, so the last thing I want to talk about in this particular section is around another approach to forming particles. So we can understand biology, and that's what we've done with looking at those biomimerization proteins, but we could also just screen a huge range of sequences, and that is the biopanning approach. So that's where we basically have a phage display library where we have different sequences displayed on phage, and the sequences vary just by one region, or two actually in this case. The variable regions are shown in these green loops in the protein on the top right here. And it's just that small area of difference that makes the whole library different, that adds the diversity. And really this is just a fishing exercise. So here we wanted to see what would bind specifically to cubic nanoparticles, cubic magnetite, and then hopefully those sequences would help form those particular particles as an additive. So the fishing exercise is we throw cubes in, we wash away everything that doesn't bind, and everything that does we know binds to cubes, and then we can obviously repeat that, so you can evolve it to get the strongest and strongest binders out. Now, interestingly, maybe you didn't spot this, but when I do talks previously around MS6, I labour the point. For those who aren't biological, D and E amino acids are the acidic amino acids. Okay, so you'll see there's acidic regions here. I talked about as well when I talked about the mimicking and MS6, they have these acidic residues. I assumed when we started doing this biopanning exercise that we'd just pull out lots of acidic sequences and say, aha, look, biology, biopanning equals the same. Actually, what we got is completely the opposite, and we pulled out a huge preference for lysine. So I've shown in blue the more basic residues. So we've got lysine and histidine and arginine here that are shown in the Ks, the Hs, and the Rs. Okay, so at different positions, this is the different position on the variable region. A huge dominance of basic residues. So the strongest binder we use as an additive again for our room temperature co-precipitation. This is the control, and when we added that additive, we get these lovely cubes. Okay, so this is controlling the formation in a way that seems very different to what's happening in biology. And actually, when we do some modelling on it, we see why. We see that the acidic residues bind ions, whereas it's the basic residues that can interact with the magnetite surface, and different surface faces will then give different morphologies of the particle itself. Okay. So as an aside, we've got a patent now on something called MagTag, where basically we realize these proteins bind very strongly to magnetite, so we could use that because they bind strongly to magnetite. So we can then make fusion proteins using the MagTag. So it's a tag that's attached to magnetite, but then we can have the fusion protein with, say, an enzyme, or indeed anything you want to isolate and purify. And this could be used for lab scale purification or even industrial scale purification with a magnet. You could use it for biologically making more drugs. That biomedicine again, it's something that one of my PhD students spent a lot of time working on, basically just using pure magnetite nanoparticles as an anchor then, and using MagTag as an anchor for many different drug products, and even immobilizing enzymes for reusability. Anyway, that was an aside. We've thought through around these different sorts of approaches to understanding the proteins, but now we can think, I think, around a pipeline. It's been sort of in the back of the talk as I've been talking about the idea of scale up, and the idea that the magnetosomes themselves aren't that difficult to produce. I've talked about the fact that the MS proteins are a bit more tricky as well. So how can we get that understanding? So we can use the proteins in vitro, we gain understanding, we can make additives, as I've already alluded to with the MMSF coil, hopefully towards scale up. Now we've learned from the magnetite interacting proteins, the aderons that we've just looked at, that actually it's the basic residues that are really important for controlling shape. And that, as I said in the very first slide around biomedicine, that's actually what gives the shape is really important for the biomedical applications. Having points and edges and facets really improves a lot of the applications. So if we're looking at scale up, maybe we should not try and look to proteins at all. Maybe we should go beyond that to very simple organic molecules. So we've got these amine residues that are really important for binding to the surfaces. So why not just screen a whole range of linear amines? So we looked at the ethylene diamine series, where basically we have 2 carbons between our amines at varying lengths. So recall, So recall that we have our protein additive. Now, if we just change that to these ethylene diamine series of additives, can we get a similar effect, learning from nature, but not actually using the proteins at all? And this is something that we've got under review at the moment in Green Chemistry, RSC journal. So here we have the series, Deta, Teta, Tepa, and Piha, which is just an increasing length of amines, and the control. And hopefully you can see that actually we get more faceted particles the more length there is on our additive. So this is the shape analysis, and you can see that actually Deta isn't the best, but once we get to Teta, Tepa, and Piha, then we really have that shape control. There isn't really much control over the size. The size seems to be similar throughout. We've done the modelling on this as well, and we see that actually, conversely to what we saw with the Mears proteins, actually the modelling preferences the 111 surface of magnetite rather than 100, and that preference means actually we make octahedral particles. There's a preference for octahedral particles rather than cubic. The process is also important. So I've mentioned having a simpler additive, but how we actually do the synthesis is really important too. So, for example, comparing batch versus flow synthesis, and then a small-scale flow synthesis we've called mini-fluidics compared to large-scale turbulent flow synthesis. We've done a lot of work in this as well. Again, some of this is waiting to be published. Some of it has been published. So we use this mini-fluidic system where we analyse some of the protein work in flow, and that was published a couple of years back, or a year and a half ago. More recently, now I've been looking at those amine additives in both these systems, the mini-fluidic and the macro-scale turbulent flow, and again, that's ready to submit that paper as well. But you can see from these results, what we do see is that there is this preference for making more angular, faceted, octahedral particles with the additives, even in large-scale turbulent flow. That's what these results are from. Okay, so I just want to end the last couple of minutes to think how we can sort of bring all this together and where we can go beyond magnetite and beyond sort of the biomedical to what was promised in the title, which is also nanotechnological applications. So cobalt platinum for data storage. So cobalt platinum, very briefly, it's really sort of ideal for data storage because you can make very, very small particles of it, and it will retain the magnetic signal, the bit of information, if it's the L10 phase. Okay, and that is basically just the ordered phase. So you've got this layered structure of cobalt platinum layering up compared to the disordered structure. The problem is, and the reason why it isn't used so much, is because to get that ordered phase, you need to do some very high temperature annealing, and that will make the particles aggregate and cause all sorts of problems. And also, it's not very green. So I'm not the first person to think that actually wouldn't it be good if we could get biology to try to organise and help form these particles in the right structure. And indeed, from a long way back now, from 2004, this paper, different groups, Trevor Douglas, Angela Belcher, have found cobalt platinum controlling peptides and also iron platinum controlling peptides. What we thought we'd do is we thought we'd analyse these through the spot array technique, where basically we can vary the sequence in a very systematic, controlled manner to try to understand if there's one particular residue. So we can have aniline scanning, where we just take out each residue in turn and then replace it with something very bland. We can truncate the sequence, so we can see if there's chunks of motifs or length that's important, and we can look for lead peptide sequences and high frequency scanning. So if a certain residue pops up a lot, we can scan against those as well. And importantly, we did this against the reagents during the synthetic process and on the products as well. So we can find lots of all the important things. And because I'm running out of time, I think it's important to kind of just give you the key findings. And what we found is the importance of basic residues. So lysine, again, we're seeing this a bit of a theme, aren't we, from the MERS proteins as well. Lysine was really important, especially at the end, so at either ends. The importance of methionine popped up as well. And there were a few sequences that were favoured over others, as well as shorter sequences. So from these findings, we thought, well, why not try to design, so we can basically, we've got the biopanning process, we can understand it, we can iterate it, and now we can try to design from it. So that's exactly what we did. We designed some amino acid sequences, and apologies, I'll have to tip your head on your side here. This is that lead one, the last one here in green, that was very good. But then we popped in a methionine, swapped out the acidic residues that didn't seem to be very helpful at all, and put a methionine in, and we've seen there's an increase in hardness, which is obviously that L10 coming through. Similarly, we tried popping methionine into different locations and then made shorter ones, and you can see that when we then substituted in the two key motifs and then popped in a methionine as well, we could basically design from the understanding that we'd got the best quite short peptide. So I end by thinking about the future, and I'm basically kind of proposing this pipeline where we can identify any nanoscale inorganic material, not just magnetic, and we could biopan against those materials. We could optimize that biopanning through this spot array technique. We can understand it more, and then perhaps we could design additives. So there's my conclusions. It's very wordy, you can read it at your leisure. But basically, just to remind you that the magnetosomes are excellent material for nanomedicine, and they can be enhanced. The magnetosomes can be inspiration to create biomimetic magnetosomes, both magnetoliposomes and magnetopolymersomes. The proteins can be used to form particles in vitro, and as well artificial proteins have been found from biopanning to control shape, and we can substitute those for bio-inspired particles. Lots of people to acknowledge. I won't read them all because we've run out of time, but multidisciplinary work it's essential to work with lots of people, and I thank everyone here, and probably lots of people I've missed out as well, for all their input into this work, particularly our group members.